Hello dear students, welcome back to GDC. In this video, we are going to decode one important topic, that is the classification of carbohydrates. So students always face problem to remember the classification of carbohydrate. And if you remember, there are many questions has been asked in GPAT in recent few years. So this is one of the important topic which we are going to decode today. So first is, what is carbohydrate? Let's start. So let's decode one by one. So first is, what is carbohydrate? So carbohydrates are nothing but they are hydrates of they are hydrates of carbon. Okay, they are hydrates of carbon. So they are basically consisting of three major organic component. One is your carbon, one is your hydrogen, one is your oxygen. CHO. They are consisting of carbon, hydrogen, and oxygen. So carbohydrates are the major source of energy provider to our body. When we heard the word carbohydrate, that means we always recall about energy. Okay, so if you remember during the sports activities during our school time, after the running or after any athletic event, our teachers used to feed us glucose. So why? Because this is because it provides us instant energy. Okay, so when we heard this word carbohydrate, you always recall about what? Energy. Okay, we always recall about energy. Now, so question is, how much energy we will get from 1 gram carbohydrate? So please remember, from 1 gram carbohydrate, we will receive around 4 kilocalorie of energy. From 1 gram carbohydrate, we will basically receive around 4 kilocalorie of energy. Okay, now let's discuss each and every point in detail. So first thing is, carbohydrates are nothing but they are polyhydroxyaldehyde and ketone. They are polyhydroxyaldehyde or ketone. Okay, polyhydroxyaldehyde and ketone. So they are consisting of aldehyde group, along with that ketone group, aldehyde group and ketone group. Another word is polyhydroxy. Poly means more number, hydroxyl means this hydroxyl. So they are consisting of multiple units of OH group along with this aldehyde and ketone group. Okay, and these groups are always combined to provide us energy. Okay, now so this is basic information we have discussed. Now, general formula. So carbohydrates are basically divided into three major categories. One is your monosaccharide, second one is your disaccharide, and third one is your polysaccharides. Okay, so these monosaccharide, let's talk one by one. This monosaccharide, okay, this monosaccharide, if we'll talk, so these monosaccharides are nothing but these are the simplest sugar. What are they? They are simplest sugar. And this is having a general formula CnH2nOn. CnH2nOn. Some book you will find like CH2O N. Okay, both are meaning same. So in GPAT 2023, one question was asked about what is the ratio of hydrogen to oxygen. So how you remember? So if this formula basically talks about the number. So here carbon 1, hydrogen 2, and oxygen 1. So ratio is 1 is to 2 is to 1 for what? 1 is to 2 is to 1 for this monosaccharide. Okay. Now, so what here N stands for? You are finding N. So what this N basically stands for? N basically stands for number of carbon. N stands for number of carbon. Okay, carbon, hydrogen, oxygen, and here N stands for number of carbon. Okay, now is classification. How we are going to remember the classification? So if you'll see, so that is a simple mnemonic that try to play handball happily. This is the mnemonic. Okay. Try to play handball happily. So before going to discuss, let me tell you one more thing. So these monosaccharides are further divided into two classes. One is called aldose, another one is called as ketose. One is aldose, another one is your ketose. Aldose means what? These are the class of compound which contains aldehyde as a functional group. That's why it is called as aldose. What is ketose? They contain ketone as a functional. Okay, this is the difference between aldose and ketose. In case of aldose, aldehyde is present as a functional group. In case of ketose, ketone is present as a functional group. This is very clear, simple terminology you should remember. Let's T stands for what this try to play handball happily stands for. Let's decode this. Try, try stands for triose, tetrose, pentose, hexose, and heptose. So triose means here n equal here c n equal to 3. Here n equal to 3. So formula will be C3H6O3. Here tetrose means n equal to 4. That is C4H8O4. Pentose n equal to 5. That is C5H10O5. Hexose n equal to 6, that is C6H12O6. Next is heptose, that is 7, n equal to 7, that is C7H14O7. This is the general formula for this monosaccharide. Now the thing is how we will going to remember this. Okay, how we will remember. So for the remembering example, let's see what are the example. Triose, you have two example. One is your glyceraldehyde. Okay, another one is your dihydroxyacetone. We will discuss when we will talk about glycolysis because these two is intermediate in glycolysis. Next is tetrose. You have example is erythrose and erythrulose. In case of tetrose, example is erythrose and erythrulose. Next is pentose. What is the example of pentose? So in pentose, you have 5 different examples. One is your ribose, ribulose, deoxyribose, xylose, and your xylulose. These are comes under your pentose sugar. Now we'll talk about hexose sugar. Hexose means 6. So there is a mnemonic. The mnemonic is good girl makes fun. So hexose example is good girl make fun. That is your glucose, galactose, mannose, and your fructose. Okay, glucose, galactose, mannose, and fructose. Now, when I will take you another lecture of decode series, that is isomerism part. I will explain you very clearly about the isomerism of carbohydrate. In that isomerism topic, one topic is concept of epimer. So when you will find glucose and galactose are of C4 epimers, and glucose and mannose are C2 epimers. Glucose and galactose are C4 epimer. Glucose and galactose are C2 epimer. Now, what is epimer basically? So, if I will tell you just briefly, epimers are what? So, these are the class of compound, or these are the monosaccharide. They are differ from each other. Okay, these are the two monosaccharide. They are differ from each other. How they will going to differ from each other? They are differ from each other around a single carbon. If you will compare the structure of glucose and galactose, they are differ from each other at carbon number 4. Similarly, if you will find out what is the difference between glucose and mannose, you will find their difference is at carbon number 2. Okay, these are the examples. And what we have discussed, these are the example of your hexose class. And if we talk about the heptose class, heptose, the example is D-pseudoheptulose. And please remember, these D-pseudoheptulose are basically act as a intermediate. It is act as a intermediate in your HMP salt. Intermediate in HMP salt. Okay, this is the example of heptose. Now we will talk about, we have completed the monosaccharide. Now we will talk about what? We will talk about oligosaccharide. So oligosaccharide means when it contains 2 to 10 monosaccharide. When it contains 2 to 10 monosaccharides, we are called it as a oligosaccharide. Now, these oligosaccharides are further divided into two subclasses. One is called as disaccharide, another is called as trisaccharide. The disaccharides are nothing but, so how you will remember the example of disaccharide? You have to remember the simple mnemonic Suleiman. What you have to remember? You have to remember the mnemonic Suleiman. So how you will remember from Suleiman? That is sucrose, lactose, maltose. Sucrose is called as, yes, cane sugar. Sucrose is also called as invert sugar. Okay. Lactose is also called as milk sugar. Now the thing is, as we are talked about, this is a disaccharide. So disaccharide means it should consisting of minimum 2 monosaccharide. So what are the monosaccharide to is prepared? Let's discuss. Maltose consisting of alpha D glucose plus beta D glucose. Okay. Lactose is consisting of glucose plus galactose. When glucose and galactose combine, it will give lactose. Similarly, when glucose plus fructose, they combine, it will give sucrose. Okay, these are the example of disaccharide. Now, again is trisaccharide. Trisaccharide means they are consisting of 3 monosaccharide. So trisaccharide example 1 is your raffinose. Okay. Another one is your xanthinose. Okay. We are discussing about, we are completed about raffinose, oligosaccharide. So oligosaccharide consisting of 2 major monosaccharide. One is your Oligosaccharide consisting of two types, disaccharide and trisaccharide. In case of disaccharide, the example is your sucrose, lactose, and maltose. In case of trisaccharide, example is your raffinose and xanthine. Okay, so this is the example of what? This is the example of oligosaccharide. Now the example is polysaccharide. What is polysaccharide? Polysaccharide means when it is consisting of more monosaccharide unit, we call it as a polysaccharide. Again, these polysaccharides are subdivided into two major class. One is called homopolysaccharide. Another one is called as heteropolysaccharide. One is called homopolysaccharide. Another one is called as heteropolysaccharide. So homopolysaccharide means what? Those are the polysaccharide which is consist of single unit of carbohydrate, a single type of carbohydrate. So to remember, there is a simple mnemonic. The mnemonic is GDC is super. So you know na, our GDC is so super. We are bringing so many informative content for all of. Okay, so we deserve a likes and subscribe for this video. So I hope all of you have subscribed our channel. So G stands for glycogen, T stands for dextrin, C stands for cellulose, I stands for inulin, A stands for starch. So starch and glycogen, please remember, starch is the storage form of glucose in plant. Glycogen is the storage form of glucose in animal. Okay, starch is the storage form of glucose in case of plant. Glycogen is the storage form of glucose in case of animal. Many times this question was asked. Another question always asked about this inulin. What is inulin? Never confuse this inulin with insulin. Inulin is a homopolysaccharide which is used to measurement of your glomerular filtration rate. Why it is used? It is used to measure your GFR rate. Okay. Now next one is your heteropolysaccharide. What is heteropolysaccharide? Heteropolysaccharide that they are consisting of different type of monosaccharide units that are called as heteropolysaccharide. To remember this heteropolysaccharide, you should remember a simple mnemonic, okay, so that you can remember all the name of this heteropolysaccharide. That is king, king, call his department head. Okay, mnemonic is king, call his department head. So K stands for keratin sulfate. Okay. C stands for chondroitin sulfate. H stands for heparin. D stands for, okay, D stands for dextran sulfate. Sorry, dormatan sulfate. D stands for dormatan sulfate, and another H stands for hyaluronidase. Okay, this is the example of heteropolysaccharide. So we have discussed the example of monosaccharide. We have discussed the example of disaccharide or trisaccharide. We have discussed the example of polysaccharide. So in this decode series, what we decode? We decode what is carbohydrate, how this carbohydrate looks like, what is its chemical structure, what is its classification, what is its example, and what are the mnemonics, which is the I have shared with you to remember this example. Okay, I hope up to this it is clear to all of you. Now let's see what are this function of this monosaccharide and disaccharide. Okay, so first is your glyceraldehyde. So glyceraldehyde and dihydroxyacetone, these are the two intermediate of glycolysis. Okay, so question is asked, what is glycolysis? Glycolysis is nothing but this is the oxidation of glucose to pyruvate or lactate depending upon the presence or absence of oxygen. And during this oxidation of glucose, there is a production of ATP which is going to be utilized through the different metabolic process of our body. Okay, next is erythrose. Erythrose is basically it is an intermediate in carbohydrate metabolism. Ribose and deoxyribose. Ribose and deoxyribose are the sugar present in nucleic acid. In exam, question always ask about what is the difference between ribose. Two question it can be asked here. So what is the difference between D-ribose? Or question is like, what is the difference between ribose and deoxyribose? Here, two questions can be asked. One is what is difference and what kind of difference. So what is the difference between in case of ribose, in carbon number 2, you will have this OH group. But in case of deoxyribose, this OH group is replaced with hydrogen. Here, this O is removed. That's why deox means removal of this oxygen. Okay. So what is the replacement? So OH is replaced by H. At what position? The position number is carbon number 2. Question is asked. So I have shared you. Please remember it can be asked in your exam. Okay. Next is ribulose. Ribulose is basically produced as a metabolite in HMP shunt. Xylose is a component of glycoproteins. Xylulose is basically one of the important marker in pentosuria because it is excreted in urine. D-lyso, this is a constituent of heart muscles. Next is glucose. Glucose is basically considered as the sugar fuel of life. Galactose is basically component of this galactosemia. Mannose is component of polysaccharide. Fructose is basically present. This type of fructose is basically present in fruit. Okay, this is also called as fruit sugar. Okay, please remember. And pseudoheptylose, this is one of the intermediate in HMP pathway. Next is your disaccharide. One is your sucrose, lactose, and maltose. Lactose is called as milk sugar. Lactose is called as milk sugar. Maltose is produced as a hydrolysis of starch. Sucrose is a constituent of your cane sugar. Okay, you remember this. So what are the biochemical functions of this disaccharide? Okay, I hope up to this everyone is clear. So we have discussed here about the classification of monosaccharide carbohydrate, that is monosaccharide or disaccharide, trisaccharide, and polysaccharide. In polysaccharide, again, we have discussed about homopolysaccharide and heteropolysaccharide. Okay, we have discussed about. Now, the next things we are going to decode is let's practice some MCQ. So, if we'll practice some MCQ, it will make a clarity to us so that our concept will be clear. Okay, so let's discuss. So, first question is carbohydrate have a hydrogen to oxygen atom ratio. Okay, so remember that, recall that formula, that is CnH2nOn. Carbon is 1, hydrogen is 2, oxygen is 1. So 1 is to 2 is to 1. Now question is carbohydrate have hydrogen to oxygen ratio. That is hydrogen is 2 and oxygen is 1. That is 2 is to 1. So option D is the correct answer. And if the question is about carbon to hydrogen ratio, then the answer will be 1 is to 2. If the question is about carbon to hydrogen ratio, the question was here is hydrogen to oxygen atom ratio. Okay, option D is the correct answer. This question was asked in GPAT 2023 SIP 2. So whatever question I am discussing with you has been asked in some of your important exam, not only in GPAT, some question are also asked in different DI exam. Okay, so you should remember, okay, what are the concept behind the question. Next question, which of the following is a triose? Triose means N equal to 3. So formula will be C3H6O3. Okay. So if you remember triose, you have two compounds. One is your glyceraldehyde, another one is your dihydroxyacetone. In glyceraldehyde is a aldose. Glyceraldehyde aldose that is having aldehyde functional group. Dihydroxyacetone is a ketose having ketone functional. Okay, you should remember what is aldose and what is ketose. Okay, now here the correct answer is option D. Let's see why these three options will not be the answer, because glucose is a hexose. Ribose and ribulose both are a pentose. Okay, that's why the option is not correct. I hope it is clear to all of you. Now, next question. Which of the following disaccharide? Which of the following is a disaccharide? So disaccharide, for disaccharide, what is the mnemonic I told you? I told the mnemonic is Suleiman. I tell the mnemonic Suleiman. That is sucrose, lactose, maltose. So here lactose is there. So option B is the correct. So why not dextrose? Because dextrose is nothing but dextrose is a dextrorotatory. This is a nothing but this is D-glucose. I will tell you what is D-glucose, what is L-glucose. D-glucose means dextrorotatory form of glucose. Okay. Next is uh, glucose. Glucose is again hexose. This is a monosaccharide. Fructose, again it is a hexose. This is also a monosaccharide. I hope it is clear to all. The mnemonic is Suleiman. Okay, the mnemonic is Suleiman. Next question. Starch is. This question was asked in Bihar DI 2020. So if you remember, I have told you two mnemonics to remember polysaccharide. Okay, for polysaccharide, I tell two mnemonic. One for homo, one for hetero. For homopolysaccharide, what is the mnemonic? GDC is super. So G stands for glycogen, D stands for dextrin, C stands for cellulose, I stands for inulin, S stands for starch. Okay, so starch is a homopolysaccharide. No, it is not a disaccharide, not a monosaccharide, not a mucopolysaccharide. Okay, so and starch, what is starch basically? Starch is nothing but this is a storage form of glucose in plant, and glycogen is a storage form of glucose in case of animal food. Storage form of glucose in case of animal. Okay, I hope it is clear to all of you. No doubt at all. So what, let me summarize what we have learned in today's class. So in today's class, we talked about basic carbohydrate, what is its function, what are the, I told you carbohydrates are polyhydroxyaldehyde and ketone. Okay, they contain more group of aldehyde and ketones. Next, we talked about this, what is its formula, that is general formula of carbohydrate. Then we talked about the classification of monosaccharide, that is triose, tetrose, pentose, hexose, heptose, and we also deal with the example. Okay, we also deal with the example. Okay, glyceraldehyde, dihydroxyacetone, we have discussed. Next, we discuss about the classification monosaccharide. You have triose, glyceraldehyde, dihydroxyacetone, erythrose, erythrulose, ribose, ribulose. Okay, hexose, you have glucose, galactose, mannose, and fructose. Heptose, you have deacetohexose. Next, in oligosaccharide, you have sucrose, lactose, and maltose. In trisaccharide, we have raffinose and xanthionose. Next, in case of polysaccharide, we have homopolysaccharide and heteropolysaccharide. Homopolysaccharide example is a starch, dextrin, inulin, glycogen, cellulose. In heteropolysaccharide example is hyaluronic acid, chondroitin sulfate, heparin, keratin sulfate, or others. Next, we talked about the mnemonics. Okay. Next, we talked about the classification with mnemonics. We have talked about the functions. And at last, we have discussed steps. I hope all of you have enjoyed this session a lot. If you have any question, okay, you directly put your question in the comment section. We will answer your each and every question. And try to give a feedback after this video how you like this video. Thank you. Thank you so much.